43.3 Human reproductive anatomy and gametogenesis. By the end of this section, you will be able to do the following: describe human male and female reproductive anatomies, discuss the human sexual response, describe spermatogenesis and oogenesis, and discuss their differences and similarities as animals became more complex. Specific organs and organ systems developed to support specific functions for the organism. The reproductive structures that evolved in land animals allow males and females to mate, fertilize internally, and support the growth and development of offspring. Human reproductive anatomy. The reproductive tissues of male and female humans develop similarly in utero until a low level of the hormone testosterone is released from male gonads. Testosterone causes the undeveloped tissues to differentiate into male sexual organs. When testosterone is absent, the tissues develop into female sexual tissues. Primitive gonads become testes or ovaries. Tissues that produce a penis in males produce a clitoris in females. The tissue that will become the scrotum in a male becomes the labia in a female. That is, they are homologous structures. Male reproductive anatomy. In the male reproductive system, the scrotum houses the testicles or testes, singular. Testis, including providing passage for blood vessels, nerves, and muscles related to testicular function. The testes are a pair of male reproductive organs that produce sperm and some reproductive hormones. Each testis is approximately 2.5 by 3.8 centimeters, 1.5 by 1 in size, and divided into wedge-shaped lobules by connective tissue called septa. Coiled in each wedge are seminiferous tubules that produce sperm. Sperm are immobile at body temperature. Therefore, the scrotum and penis are external to the body, as illustrated in figure 43.8, so that a proper temperature is maintained for motility. In land mammals, the pair of testes must be suspended outside the body at about 2 degrees Celsius lower than body temperature to produce viable sperm. Infertility can occur in land mammals when the testes do not descend through the abdominal cavity during fetal development. 43.3 Human reproductive anatomy and gametogenesis. 1231 Visual connection. Figure 43.8 The reproductive structures of the human male are shown. Which of the following statements about the male reproductive system is false? A. The vas deferens carries sperm from the testes to the penis. B. Sperm mature in seminiferous tubules in the testes. C. Both the prostate and the bulbourethral glands produce components of the semen. D. The prostate gland is located in the testes. Sperm mature in seminiferous tubules that are coiled inside the testes, as illustrated in figure 43.8. The walls of the seminiferous tubules are made up of the developing sperm cells, with the least developed sperm at the periphery of the tubule and the fully developed sperm in the lumen. The sperm cells are mixed with nursemaid cells called Sertoli cells, which protect the germ cells and promote their development. Other cells mixed in the wall of the tubules are the interstitial cells of Leydig. These cells produce high levels of testosterone once the male reaches adolescence. When the sperm have developed flagella and are nearly mature, they leave the testicles and enter the epididymis, shown in figure 43.8. This structure resembles a comma and lies along the top and posterior portion of the testes. It is the site of sperm maturation. The sperm leave the epididymis and enter the vas deferens, or ductus deferens, which carries the sperm, behind the bladder, and forms the ejaculatory duct with the duct from the seminal vesicles. During a vasectomy, a section of the vas deferens is removed, preventing sperm from being passed out of the body during ejaculation and preventing fertilization. Semen is a mixture of sperm and spermatic duct secretions, about 10% of the total, and fluids from accessory glands that contribute most of the semen's volume. Sperm are haploid cells, consisting of a flagellum as a tail, a neck that contains the cell's energy-producing mitochondria, and a head that contains the genetic material. Figure 43.9 shows a micrograph of human sperm as well as a diagram of the parts of the sperm. An acrosome is found at the top of the head of the sperm. This structure contains lysosomal enzymes that can digest the protective coverings that surround the egg to help the sperm penetrate and fertilize the egg. An ejaculate will contain from 2 to 5 milliliters of fluid with from 50 to 120 million sperm per milliliter. 1232 Chapter 43, Animal Reproduction and Development. Access for free at openstax.org. Figure 43.9 Human sperm, visualized using scanning electron microscopy, have a flagellum, neck, and head. Credit B. Modification of work by Mariana Ruiz Villarreal. Scale bar data from Matt Russell. The bulk of the semen comes from the accessory glands associated with the male reproductive system. These are the seminal vesicles, the prostate gland, and the bulbourethral gland, all of which are illustrated in Figure 43.8. The seminal vesicles are a pair of glands that lie along the posterior border of the urinary bladder. The glands make a solution that is thick, yellowish, and alkaline. As sperm are only motile in an alkaline environment, a basic pH is important to reverse the acidity of the vaginal environment. The solution also contains mucus, fructose, a sperm mitochondrial nutrient, a coagulating enzyme, ascorbic acid, and local acting hormones called prostaglandins. The seminal vesicle glands account for 60% of the bulk of semen. The penis, illustrated in Figure 43.8, is an organ that drains urine from the renal bladder and functions as a copulatory organ during intercourse. The penis contains 3 tubes of erectile tissue running through the length of the organ. These consist of a pair of tubes on the dorsal side, called the corpus cavernosum, and a single tube of tissue on the ventral side, called the corpus spongiosum. This tissue will become engorged with blood, becoming erect and hard, in preparation for intercourse. The organ is inserted into the vagina, culminating with an ejaculation. During intercourse, the smooth muscle sphincters at the opening to the renal bladder close and prevent urine from entering the penis. An orgasm is a 2-stage process. First, glands and accessory organs connected to the testes contract, then semen, containing sperm, is expelled through the urethra during ejaculation. After intercourse, the blood drains from the erectile tissue and the penis becomes flaccid. The walnut-shaped prostate gland surrounds the urethra, the connection to the urinary bladder. It has a series of short ducts that directly connect to the urethra. The gland is a mixture of smooth muscle and glandular tissue. The muscle provides much of the force needed for ejaculation to occur. The glandular tissue makes a thin, milky fluid that contains citrate, a nutrient, enzymes, and prostate-specific antigen, PSA. PSA is a proteolytic enzyme that helps to liquefy the ejaculate several minutes after release from the male. Prostate gland secretions account for about 30% of the bulk of semen. The bulbourethral gland, or Cowper's gland, releases its secretion prior to the release of the bulk of the semen. It neutralizes any acid residue in the urethra left over from urine. This usually accounts for a couple of drops of fluid in the total ejaculate and may contain a few sperm. Withdrawal of the penis from the vagina before ejaculation to prevent pregnancy may not work if sperm are present in the bulbourethral gland secretions. The location and functions of the male reproductive organs are summarized in Table 43.1. Male reproductive anatomy: organ location, function, scrotum, external carry and support, testes, penis, external, deliver urine, copulating organ, testes, internal, produce sperm and male hormones. Table 43.1 43.3. Human reproductive anatomy and gametogenesis. 1233. Organ location, function, seminal vesicles, internal, contribute to semen production, prostate gland, internal, contribute to semen production, bulbourethral glands, internal, clean urethra at ejaculation. Table 43.1. Female reproductive anatomy: a number of reproductive structures are exterior to the female's body. These include the breasts and the vulva, which consists of the mons pubis, clitoris, labia majora, labia minora, and the vestibular glands, all illustrated in Figure 43.10. The location and functions of the female reproductive organs are summarized in Table 43.2. The vulva is an area associated with the vestibule, which includes the structures found in the inguinal, groin, area of women. The mons pubis is a round, fatty area that overlays the pubic symphysis. The clitoris is a structure with erectile tissue that contains a large number of sensory nerves and serves as a source of stimulation during intercourse. The labia majora are a pair of elongated folds of tissue that run posterior from the mons pubis and enclose the other components of the vulva. The labia majora derive from the same tissue that produces the scrotum in a male. The labia minora are thin folds of tissue centrally located within the labia majora. These labia protect the openings to the vagina and urethra. The mons pubis and the anterior portion of the labia majora become covered with hair during adolescence. The labia minora is hairless. The greater vestibular glands are found at the sides of the vaginal opening and provide lubrication during intercourse. Figure 43.10. The reproductive structures of the human female are shown. Credit A. Modification of work by Gray's Anatomy. Credit B. Modification of work by CDC, Female Reproductive Anatomy. Organ location function: clitoris, external sensory organ, mons pubis, external fatty area, overlying pubic bone. Table 43.2 1234. Chapter 43. Animal Reproduction and Development. Access for free at openstax.org. Organ location function: labia majora, external covers labia minora. Labia minora, external covers vestibule. Greater vestibular glands external secrete mucus. Lubricate vagina. Breasts external produce and deliver milk. Ovaries internal carry and develop eggs. Oviducts, fallopian tubes, internal transport egg to uterus. Uterus internal support developing embryo. Vagina internal common tube for intercourse. Birth canal, passing menstrual flow. Table 43.2. The breasts consist of mammary glands and fat. The size of the breast is determined by the amount of fat deposited behind the gland. Each gland consists of 15 to 25 lobes that have ducts that empty at the nipple and that supply the nursing child with nutrient and antibody-rich milk to aid development and protect the child. Internal female reproductive structures include ovaries, oviducts, the uterus, and the vagina, shown in Figure 43.10. The pair of ovaries is held in place in the abdominal cavity by a system of ligaments. Ovaries consist of a medulla and cortex. The medulla contains nerves and blood vessels to supply the cortex with nutrients and remove waste. The outer layers of cells of the cortex are the functional parts of the ovaries. The cortex is made up of follicular cells that surround eggs that develop during fetal development in utero. During the menstrual period, a batch of follicular cells develops and prepares the eggs for release. At ovulation, one follicle ruptures and one egg is released, as illustrated in Figure 43.11a. Figure 43.11. Oocytes develop in follicles, located in the ovary. At the beginning of the menstrual cycle, the follicle matures. At ovulation, the follicle ruptures, releasing the egg. The follicle becomes a corpus luteum, which eventually degenerates. The B follicle in this light micrograph has an oocyte at its center. Credit A. Modification of work by NIH. Scale bar data from Matt. Russell. The oviducts, or fallopian tubes, extend from the uterus in the lower abdominal cavity to the ovaries, but they are not in contact with the ovaries. The lateral ends of the oviducts flare out into a trumpet-like structure and have a fringe of finger-like projections called fimbriae, illustrated in Figure 43.10b. When an egg is released at ovulation, the fimbriae help the nonmotile egg enter into the tube and passage to the uterus. The walls of the oviducts are ciliated and are made up mostly of smooth. 43.3. Human Reproductive Anatomy and Gametogenesis 1235 muscle. The cilia beat toward the middle, and the smooth muscle contracts in the same direction, moving the egg toward the uterus. Fertilization usually takes place within the oviducts and the developing embryo is moved toward the uterus for development. It usually takes the egg or embryo a week to travel through the oviduct. Sterilization in women is called a tubal ligation. It is analogous to a vasectomy in males in that the oviducts are severed and sealed. The uterus is a structure about the size of a woman's fist. This is lined with an endometrium rich in blood vessels and mucus glands. The uterus supports the developing embryo and fetus during gestation. The thickest portion of the wall of the uterus is made of smooth muscle. Contractions of the smooth muscle in the uterus aid in passing the baby through the vagina during labor. A portion of the lining of the uterus sloughs off during each menstrual period, and then builds up again in preparation for an implantation. Part of the uterus, called the cervix, protrudes into the top of the vagina. The cervix functions as the birth canal. The vagina is a muscular tube that serves several purposes. It allows menstrual flow to leave the body. It is the receptacle for the penis during intercourse and the vessel for the delivery of offspring. It is lined by stratified squamous epithelial cells to protect the underlying tissue. Sexual response during intercourse. The sexual response in humans is both psychological and physiological. Both sexes experience sexual arousal through psychological and physical stimulation. There are 4 phases of the sexual response. During phase 1, called excitement, Vasodilation leads to vasocongestion in erectile tissues in both men and women. The nipples, clitoris, labia, and penis engorge with blood and become enlarged. Vaginal secretions are released to lubricate the vagina to facilitate intercourse. During the second phase, called the plateau, stimulation continues. The outer third of the vaginal wall enlarges with blood, and breathing and heart rate increase. During phase 3, or orgasm, rhythmic, involuntary contractions of muscles occur in both sexes. In the male, the reproductive accessory glands and tubules constrict, placing semen in the urethra. Then the urethra contracts, expelling the semen through the penis. In women, the uterus and vaginal muscles contract in waves that may last slightly less than a second each. During phase 4, or resolution, the processes described in the first 3 phases reverse themselves and return to their normal state. Men experience a refractory period in which they cannot maintain an erection or ejaculate for a period of time ranging from minutes to hours. Gametogenesis, spermatogenesis and oogenesis. Gametogenesis, the production of sperm and eggs, takes place through the process of meiosis. During meiosis, 2 cell divisions separate the paired chromosomes in the nucleus and then separate the chromatids that were made during an earlier stage of the cell's life cycle. Meiosis produces haploid cells with half of each pair of chromosomes normally found in diploid cells. The production of sperm is called spermatogenesis and the production of eggs is called oogenesis. 1236 Chapter 43, Animal Reproduction and Development, access for free at openstax.org. Spermatogenesis. Figure 43.12. During spermatogenesis, 4 sperm result from each primary spermatocyte. Spermatogenesis, illustrated in Figure 43.12, occurs in the wall of the seminiferous tubules, Figure 43.8, with stem cells at the periphery of the tube and the spermatozoa at the lumen of the tube. Immediately under the capsule of the tubule are diploid, indifferentiated cells. These stem cells, called spermatogonia, singular, spermatogonium, go through mitosis with one offspring going on to differentiate into a sperm cell and the other giving rise to the next generation of sperm. Meiosis starts with a cell called a primary spermatocyte. At the end of the first meiotic division, a haploid cell is produced called a secondary spermatocyte. This cell is haploid and must go through another meiotic cell division. The cell produced at the end of meiosis is called a spermatid, and when it reaches the lumen of the tubule and grows a flagellum, it is called a sperm cell. 4 sperm result from each primary spermatocyte that goes through meiosis. Stem cells are deposited during gestation and are present at birth through the beginning of adolescence, but in an inactive state. During adolescence, gonadotropic hormones from the anterior pituitary cause the activation of these cells and the production of viable sperm. This continues into old age. Link to learning. Visit this site, http://openstax.org/l/spermatogenesis, to see the process of spermatogenesis. Oogenesis, illustrated in Figure 43.13, occurs in the outermost layers of the ovaries. As with sperm production, oogenesis starts with a germ cell, called an oogonium, plural, oogonia. But this cell undergoes mitosis to increase in number, eventually resulting in up to about 1 to 2 million cells in the embryo. 43.3, Human Reproductive Anatomy and Gametogenesis. 1237 Figure 43.13. The process of oogenesis occurs in the ovary's outermost layer. The cell starting meiosis is called a primary oocyte, as shown in Figure 43.13. This cell will start the first meiotic division and be arrested in its progress in the first prophase stage. At the time of birth, all future eggs are in the prophase stage. At adolescence, anterior pituitary hormones cause the development of a number of follicles in an ovary. This results in the primary oocyte finishing the first meiotic division. The cell divides unequally, with most of the cellular material and organelles going to one cell, called a secondary oocyte, and only one set of chromosomes and a small amount of cytoplasm going to the other cell. This second cell is called a polar body and usually dies. A secondary meiotic arrest occurs, this time at the metaphase 2 stage. At ovulation, this secondary oocyte will be released and travel toward the uterus through the oviduct. If the secondary oocyte is fertilized, the cell continues through the meiosis too, producing a second polar body and a fertilized egg containing all 46 chromosomes of a human being, half of them coming from the sperm. Egg production begins before birth, is arrested during meiosis until puberty, and then individual cells continue through at each menstrual cycle. One egg is produced from each meiotic process, with the extra chromosomes and chromatids going into polar bodies that degenerate and are reabsorbed by the body.